Protein folding kinetics is a quite complicated process, and it's actually an active field of research. So in this video, we'll show you some of the basic concepts that dictate some of the kinetics of protein folding, just to get you a sense of how protein folding happens. Remember that we already have a tool to visualize the process of protein folding, and that's the folding funnel. When we look at the protein funnel, we think of the protein starting at a high energy level when it's unfolded, and then moving down in trajectory towards the minimum. And the minimum is the native fold of the protein. But how does a protein actually change its shape as it's undergoing this folding process? Remember the hydrophobic effect? We talked about how the hydrophobic side chains will bury themselves into the hydrophobic core of protein. Well, this hydrophobic collapse is actually often one of the first steps in the folding process. When looking at these folding funnels, remember that the fraction of residues in the native conformation increases as the proteins move towards its native state. And at the same time, the number of possible conformations decreases, and that's the funnel effect here, as the side chain becomes more and more constrained. So let's dig deeper into these conformations as the protein is undergoing the process of folding to better understand the kinetics. Remember that these phi and psi bonds can rotate, but not quite freely all the way around the 360 degrees of rotation. Some of the angles are disallowed because there are some steric clashes. And we saw that the Ramachandran plot is a great way to visualize the regions that are allowed, the regions that prevent a lot of the steric clashes in proteins. And so we already know now that there's some constraints on the conformations of proteins. But the protein still needs to take on very precise dihedral angles to maintain its native fold, because just one or two errant dihedrals can actually disrupt the fold of the protein. And we can illustrate this with a simple example of an alpha helix. Remember that a series of residues within the same region of the phi-psi angles in the Ramachandran plot, the green region here, can generate an alpha helix. So say if just two residues within this alpha helix change their dihedral angle values to other parts of the Ramachandran plot, well, that would actually disrupt the whole helix. So therefore, that tells us that protein folding relies on very precise conformations of the backbone through the phi-psi angles. So now let's get an idea of the magnitude of the problem, the magnitude of the task that the protein has to go through to get to its native fold. So we're going to do a thought experiment, making some estimates about the number of conformations that the protein might be able to undertake. So we're going to take an example of a protein of 100 residues. So that's about the average size of a protein domain. So now we know that its phi-psi angles are restricted, so that we can approximate that there's roughly 3 different conformations for each residue, corresponding to the 3 different regions of the Ramachandran plot. So that would yield 3 to the 100 conformations available to the whole protein, assuming that each of these dihedral angles can behave independently. 3 to the 100 is actually an enormous number. And to get an idea of the magnitude of this number, we're going to estimate that if the protein, say, samples each of these conformations for a very short amount of time, 0.1 picoseconds, well, that would yield 10 to the 27 years to sample every single conformation. And that's orders of magnitudes longer than the lifetime of the universe. So that brings us to a paradox, right? That the protein has no time to explore all the possible conformations on the way to its native fold. This is called Levinthal's paradox, after Cyrus Levinthal, who's the first one to state this concept and this paradox. And what he hypothesized is that this paradox means that there must be some conformations that are just never explored. There must be some paths that are most common as proteins start to fold. So how do proteins then figure out which conformations might be the best ones to lead them towards their native fold? That's where cooperativity comes into play, because this cooperativity brings some residues in proximity and so it sort of makes choices for the protein. So let's see a more specific example. Remember when the hydrophobic core forms, you might have at first a contact between two residues that's labeled C1 here. But that first contact will then bring other residues in close proximity. So then a second contact that's labeled C2 here now has a higher probability of forming. So that's the cooperativity illustrated for the hydrophobic effect that leads to the collapse of the hydrophobic core. But this cooperativity is also important in forming the secondary structure elements. So let's see this example now for a secondary structure element, looking at the folding of a helix. So the largest kinetic barrier for the folding of an alpha helix is the formation of the first hydrogen bond, and that's called the nucleation event. It's analogous to when you crystallize something. The first seeds of a crystal might take a long time, but then the growth of the crystal is a lot faster. So here in an alpha helix, the first hydrogen bond might take a long time to form, but once it's formed, it brings into proximity the other residues that want to form hydrogen bonds, and then they will extend the helix by forming these hydrogen bonds on either side of that first hydrogen bond to form the complete helix. So this cooperativity is key to the assembly of secondary structure elements as well. So protein scientists who study folding have generated sort of two extreme models of protein folding to help explain it. In the first one, the secondary structure forms first. And then these secondary structure elements will then form together the tertiary structure by moving until they achieve the correct fold. And this is illustrated here as a time course with the energy on the y-axis. And you can see that first the secondary structure elements are formed and then the full fold of the protein. And this process, or this model, is called the diffusion collision model. In the other model, the hydrophobic core of the protein, that's indicated here in yellow, is what first collapses in a relatively random way. And once the core has collapsed, this allows the secondary structure elements to form because now the residues are in close proximity and the protein will adopt its native conformation. And this second model is called the nucleation condensation process. The currently available experimental results on protein folding indicate that both models can operate in protein folding, and which model is favored depends on the protein, depends on the primary sequence. So as with the thermodynamics of protein folding, the kinetics of protein folding is a process that our understanding of it is advancing more and more rapidly. And a lot of that is due to computer simulations. Now, one type of computer simulations are molecular dynamics simulations, which simulate the motion of atoms. And it's powerful enough to simulate the folding of some of the small proteins in the aqueous solution, because remember that the aqueous solution is key to the folding process. So now we're going to look at a simulation of the folding of a small protein, a 35 residue protein. It's the headpiece domain of vilin, which is a cytoskeletal protein. And this protein is actually experimentally one of the classic models of protein folding, and it's one of the fastest folding proteins that's been determined experimentally. So in the simulations, there are actually thousands of water molecules that are included in the aqueous solution, but they're not going to be shown here just for clarity. And this simulation lasts in real time just 6 microseconds. That's how long it takes for this vilin headpiece to fold. You can see that the vilin headpiece is a small fold that contains 3 helices. And they're colored here from a blue N-terminus to a red C-terminus. So this simulation starts with an extended peptide, and the peptide chain rapidly collapses onto itself. And that's similar to the nucleation condensation model of protein folding that we just saw. Then 2 helices form soon after, while that middle helix takes a lot longer to form to yield the final fold. Remarkably, the final fold is very similar to the experimentally determined structure of the headpiece. But it's important to note that this is just one sample trajectory that shows us one example of how the vilin protein might fold. And actually, every simulation of this vilin headpiece yields slightly different results, although there are themes that emerge. So protein folding kinetics are quite complicated. But we've already reached a few take-home messages, some key ideas. And one of them is that there's going to be a limited number of paths that the protein's actually going to take to reach its fold. That we've come about through looking at the Leventhal paradox and how a lot of these conformations are probably not energetically accessible. So the idea is that the landscape of the energetics of protein folding is what's going to dictate the path towards folding. And the other important idea is that the cooperativity of the process is also really important. So the cooperativity will sort of dictate which steps to take along the way, one after the other. And so that'll ensure that the protein folds in a timely manner.